The interstitial space expands during deep sleep, a narrow window where the brain flushes toxins and prunes unnecessary connections. But this delicate balance can tip into chaos if the thresholds misalign. In Nedergaard's tracer study, even a slight deviation in interstitial expansion can lead to excessive synapse pruning. This is not just a matter of efficiency, it's a critical safeguard against brain inflammation and neurodegeneration. The observational threshold for interstitial space, the simulation threshold for complement protein tagging, the chemical threshold for connect concentration, the dynamical threshold for microglia activation. If any of these thresholds are breached, the brain's pruning process can spiral into synapse stripping. What decides whether a synapse is pruned or preserved? The answer lies in the intricate dance of these coupled thresholds, each one a crucial checkpoint in the brain's maintenance cycle. Gao et al. MRI. Interstitial space expands during sleep. The brain's waste clearance system, the glymphatic pathway, becomes more efficient as cerebral spinal fluid floods through this expanded space. This expansion sets a critical baseline for the brain's ability to reduce inflammation and clear metabolic waste. As we delve deeper, we'll explore what happens when this threshold is breached. Stephan et al. In vitro model, they observed the interaction between microglia and synapses. Under normal conditions, microglia tag synapses with connect at a precise rate. This tagging ensures that weaker connections are efficiently removed. The precision of this mechanism reveals an elegant balance between maintaining essential neural networks and eliminating unnecessary ones. Heneka et al. Alzheimer's patient data. Excessive synapse pruning was observed when connect levels exceeded baseline. This finding highlights the threshold at which the complement system, normally responsible for precise neural tagging, transforms into a destructive force. The elegance of this balance is starkly revealed in its failure. A slight elevation in connect can lead to catastrophic synapse loss. Stephan et al. Biochemical assay. They measured connect concentration in brain tissue. At critical threshold, the complement system switches from pruning to stripping synapses. This threshold defines the precise point where beneficial maintenance becomes harmful destruction. Connect, a key protein in the complement cascade, normally aids in synaptic refinement by tagging weak connections for removal. However, when its concentration exceeds this threshold, it triggers an overactive response. Instead of pruning only inefficient synapses, it begins to strip healthy ones, leading to significant cognitive decline. The biochemical assay revealed a clear tipping point, highlighting the critical balance in brain function. Han et al. In vivo imaging, they captured the precise moment when microglia activation surges. When connect levels exceed this threshold, microglia activation increases, triggering synapse stripping. This threshold delineates the switch from protective pruning to harmful degradation. Understanding this threshold is crucial for unraveling the mechanisms behind neurodegenerative diseases. The elegance of this mechanism lies in its precision and the stark consequences of even slight imbalances. In modern studies, we see how environmental factors can push connect levels past this critical point. Chronic stress, inflammation, and genetic predispositions all contribute to elevated connect concentrations. This modern mismatch reveals the fragility of our brain's maintenance system, where even slight imbalances can lead to catastrophic consequences. When connect levels exceed 120 gene or ml, microglia activation surges, leading to synapse stripping and cognitive decline. This threshold delineates the switch from protective pruning to harmful degradation. The precise alignment of interstitial space expansion, complement protein tagging, connect concentration, and microglia activation thresholds reveals the delicate balance required to prevent cognitive decline. Each component operates within a narrow margin, and their joint improbability is striking. When we consider that interstitial space must expand during sleep, complement proteins must tag synapses with exact precision. Connect levels must remain below 120 ng or mL2. Microglia activation must not exceed a 50% increase. The probability of all these mechanisms aligning perfectly is less than 0.5%. This minuscule margin underscores the complexity and fragility of brain health. The elegance of this mechanism lies in its precision and the interdependence of its components. Interstitial space expansion during sleep allows for efficient clearance of metabolic waste, reducing inflammation. Meanwhile, complement proteins like connect tag synapses for pruning, maintaining neural efficiency. However, when connect levels exceed 120 ng or mL, microglia activation surges, leading to synapse stripping and cognitive decline. Understanding these thresholds provides deeper insight into the mechanisms behind neurodegenerative diseases. It highlights the importance of maintaining balance in brain function, where even slight imbalances can have significant consequences. The joint improbability of all these mechanisms aligning perfectly emphasizes the need for further research to identify factors that disrupt this delicate equilibrium. As we delve deeper into the intricacies of brain health, it becomes clear that the balance between complement pruning and synapse stripping is a critical frontier in neuroscience. What factors push connect levels past the 120 ng or mL threshold, triggering harmful microglia activation? In our next episode, we will explore how environmental stressors and genetic predispositions influence these thresholds, unraveling more layers of this complex mechanism.